So I'd like to show you how lysozyme works. Lysozyme works by cleaving the glycosidic bond between NAM and NAG that makes up the peptidoglycan layer in both gram-negative and gram-positive bacteria. However, lysozyme can only get to the peptidoglycan layer of gram-positive bacteria. So I want to go and show you why. So if the human body secretes lysozyme, it can immediately get to the structure on the surface, which is peptidoglycan of a gram-positive bacterium. In contrast, the lysozyme enzyme is very big, and there is a membrane, an outer membrane above the peptidoglycan layer in gram-negative bacteria. Therefore, the gram-negative bacteria will be resistant to lysozyme because a big enzyme cannot get past the outer membrane. It protects the peptidoglycan layer. In contrast, the gram-positive bacterium has a layer of peptidoglycan that is susceptible to lysozyme. If you're wondering, the technical name for NAM is N-acetylmuramic acid, and the technical term for NAG is N-acetylglucosamine. And what we see here attached to the N-acetylmuramic acid are amino acid peptides. So there's a peptide chain here, another peptide chain here, and then they're connected by cross-links between the, so this peptide chain is attached to this peptide chain, and that's the cross-linking that we see in the peptidoglycan layer. So the next thing I would like to talk about is the acute inflammatory response. And this response is very important, and it really evolved to function locally in a tissue where there's localized damage. So for example, if you have a splinter penetrating the skin and breaking the skin, a splinter would have microorganisms on it. Everything in the world is coated with microorganisms. Our bodies, every object that you could possibly touch. And here we see the splinter has brought in microorganisms under the skin. And what we have under the skin are macrophages. And macrophages will detect the microorganisms you see over here. And basically, as they phagocytose the bacteria, recognizing perhaps LPS for gram-negative or peptidoglycan on the surface of a gram-positive, they will then secrete chemokines, which are chemicals that help to bring in other immune cells to the area. Some of these chemokines are vasodilators, which are going to help open or increase the diameter of the capillaries so that it is easier for more white blood cells to extravasate and basically leave the capillaries by jumping in between the little spaces between the endothelial cells lining the capillaries. And furthermore, capillaries are going to express little special proteins that the macrophages and other white blood cells can use to attach to the side of the endothelium of the capillaries to then attach, and so that they can secondly leave and extravasate. So what happens is the additional white blood cells will come into the area to help attack the bacteria and eliminate all the bacteria that have been entered the body. I want to show you a little bit more about the vasodilation of the capillaries in a tissue under the acute inflammatory response. So here we have the undilated capillaries, and here we have the cross-section of the dilated capillaries. And then we look at a cutout of this, one of these capillaries on the side. Here it is being not vasodilated, and here it is after vasodilation. And I want to point out that these intercellular junctions actually physically separate, leading to increased leakage of fluid and solutes out of the vasodilated capillaries. And those little spaces are what the white blood cells can actually extravasate and leave the capillary to get into the tissue and enter the interstitial space. So I'd like to now move to the molecular level about what happens during the acute inflammatory response. So when a splinter breaks through the skin, the damaged tissue that had been damaged by the splinter coming in will secrete a molecule called bradykinin. This will then signal mast cells, which are a special type of phagocytic cell in the vicinity, to degranulate and release the molecule histamine. Histamine then can stimulate the blood vessels to vasodilate. And as the blood vessels vasodilate, we're going to have some increased amount of plasma leaving the capillaries to enter the interstitial fluid. We'll also have platelets being released into the interstitial fluid as well, and prostaglandin. Now, prostaglandin is a molecule that can stimulate itching and pain. So basically, when you have an injury, this response of the bradykinin triggering mast cells, and then the mast cells trigger the production of, they produce histamine, and then the histamine opens the blood vessels, and then we have increased fluid entering the tissue that can lead to edema or swelling. And then we also have prostaglandin molecules that are going to be producing itching and pain. So I'd like to point out there are 2 over-the-counter medicines that we can take that can inhibit steps of the acute inflammatory response. One is antihistamines. Antihistamines, of course, will reduce the function of histamine as a vasodilator. And then pain relievers like aspirin or ibuprofen can inhibit prostaglandin production and then stop the pain, reduce the pain and itching. So basically, those 2 over-the-counter drugs working together can lead to less pain and also less inflammation and edema and swelling in an area where there has been tissue damage. So phagocytosis is the process where a microorganism, such as the bacterium, can be brought into an endosome, which we call a phagosome in a macrophage. The phagosome carrying the bacteria can be fused with a lysosome, which, as you know, has degradative enzymes and reactive oxygen species that can help to destroy a microorganism. After the phagosome and lysosome have fused, the microorganism would be destroyed and killed. The immune system always has to identify what is a non-self cell and a self cell, because we don't want the immune system attacking our own cells, right? That would give us an autoimmune disease. We just want to have the immune system attacking foreign cells. So fortunately, our bodies have a protein on their surface called CD47, and CD47 is basically a stop sign for phagocytic cells to say, you are not going to perform phagocytosis on me because I'm one of your cells, right? I'm one of the cells in the human body. So CD47 prevents phagocytosis. That is why typically macrophages and other phagocytic cells in the body do not phagocytose other cells in our body. They will specifically phagocytose microorganisms that lack CD47. And I also want to introduce a new term now, which is opsonization. An antibody is called an opsonin, and there are other opsonins like complement, which we'll talk about as well, that these can bind to the surface of a microorganism and facilitate phagocytosis. So basically, the phagocytosis of a bacterium just with peptidoglycan or lipopolysaccharide on the cell surface would be less efficient than the phagocytosis of a microorganism with antibody on it. So opsonins lead to opsonization, which is enhanced phagocytosis. And here I just highlight that the phagocytosis process can basically lead to the destruction of the microorganism. There's reactive oxygen species and also degradative enzymes in the lysosome that completely will destroy the microorganisms in a phagolysosome. Another important protein of the innate immune system is the membrane attack complex of complement. The membrane attack complex is a series of several proteins that have the ability to insert into the outer membrane of bacteria and make a hole, basically a donut hole in the membrane. And this donut hole has the ability to kill gram-negative bacteria. The gram-positive bacteria are resistant to the membrane attack complex because they have peptidoglycan blocking the complement proteins from getting to the plasma membrane. In gram-negative bacteria, they have the outer membrane, and then the outer membrane is accessible to the membrane attack complex. So for gram-positive bacteria, what complement can do is it can act as an opsonin. An opsonin is a protein that can enhance phagocytosis. So I showed you that antibodies can act as an opsonin, and now I'm showing you that complement can act as an opsonin to enhance phagocytosis. So for gram-positive bacteria, the only thing that complement can do is it can enhance phagocytosis of the bacterium. For gram-negative bacteria, either complement can undo the membrane attack complex and make holes in the bacterial membrane, leading to bacterial death, or the complement can actually also induce the opsonization, which is enhanced phagocytosis of the gram-negative bacteria. And here I just want to show you the phagocytic process again, right? The bacteria enters the phagosome, fuses with the lysosome, and then gets destroyed. Those are two things I showed you, and it's really important for bacteria to think about, is the bacteria gram-negative or gram-positive? Because the immune system uses very different responses for gram-negative versus gram-positive bacteria. And for viruses, we have a whole another set of innate immune responses that can be done. So one of these is the production of interferon. So here we have a diagram of a normal cell being infected by a virus. As the normal cell gets infected by a virus, the virus is going to hijack this cell cycle of the human cell to have it now produce viral particles instead of doing whatever the human cell would normally do. So our bodies and our cells have evolved a mechanism to warn other cells that this cell has been hijacked and made into a virus factory. So while the virus is hijacking the cell cycle of the cell, this cell is going to activate the transcription of the genes to make interferon mRNA. The interferon mRNA will be translated to produce interferon, which will be secreted. It's a protein to be secreted. The interferon peptides will be released, and then this is going to warn neighboring cells that there's a virus in the body. This neighboring cell has receptors for the interferon. The interferon is going to, as it binds this receptor, it's going to initiate a signal transduction cascade, and this is going to activate transcription of genes that are needed, and then these genes will then be translated to make two very important proteins. One is an oligo A synthetase, and the other is an inactive protein kinase.